I am really happy to be here to be talking about first in human and phase 1 drug development. There is a lot that happens in this phase of development. But before we dig into things, I'd like to take care of a couple housekeeping things. First of all, many of you may have noticed that you are able to unmute yourselves. I might ask that you go ahead and mute yourself right now so we don't get any unneeded interruptions. I would also say that for each of the talks, why don't you hold your questions until after the speaker has finished? We've tried to allow for at least 5 to 10 minutes of questions at the end of each talk. So if you can hold those for that, that would be great. Alternatively, one thing you can do, you may see in the lower right-hand corner a chat window. That's a great place to write down your questions in there. So I'm looking forward to a really lively discussion after each of these. So without further ado, without further ado, let's see if I can go here. There we go. This is our overall map of the drug development phases. And I think the thing that you can see, there's a fair amount that's happening early on in discovery and preclinical, but there's a lot that really starts in phase 1, and that's what we're going to be talking about today. So it's really, basically this is the place where we start, of course, testing first in testing a drug in humans. Frequently those will start with single and multiple ascending dose studies. Formulations usually aren't final by the time we start clinical development. And then there's a number of other aspects that need to be characterized during this part of the drug development process. Key among these is really the translation. This is really the first sort of translation from preclinical information that we can gather, which can be really quite detailed, and into the clinical setting. And so basically, many of the organs that we're able to study directly in our preclinical models, rats, mice, monkeys, dogs, etc., we kind of have to infer from relatively simple measurements that we can make in humans. So the first thing that we always ask in phase 1 protocol, in a first in human protocol, usually it might be a single ascending dose study, is how much can we give safely? So identifying a maximum tolerated dose is one of the key aspects of things. And then the next piece is really a more scientific question, which is, how much is actually, what's the exposure? What are the pharmacokinetics? How does the drug change over time? And is it linear with dose, for example? And then the next really critical question is not just how long does it hang around, but does the drug engage with its target? So typically we're looking for biomarker information there, and maybe even some aspects of whether the drug has some efficacy or not. And that would be primarily when we need to conduct phase 1 in a patient population. Later in development, phase 1 really, we always kind of look at phase 1 as being a distinct phase of development. Then once that's finished, then we go on to phase 2. But the reality is that the clinical pharmacology phase 1 type study continue all the way through development. So we have to always find out, so what affects the drug exposure? So we look at food, of course, physiological impairment. Typically, this would be renal and hepatic impairment. And then other aspects of things. We might do dedicated studies to understand gender differences or geographic differences or ethnicity differences, and for age differences, depending on what the indication for the drug is. Another key aspect that we would be exploring pretty typically would be effects of formulations. Typically, the first thing that we are able to study is not the formulation that would end up being marketed. So we need to understand, the bioequivalence aspects, and typically that will be a small study. And then finally, drug-drug interaction studies are things that we would do pretty frequently. So there's a whole range of activities that are occurring in this phase of development. And luckily, we have a number of speakers today who are going to address all of those things. We're going to start off with Monika Trarogovska, and I hope I said that more or less right, who is Applications Manager with Simulations Plus, and she's going to talk about, and she works in our Lixoft division in Paris, so primarily responsible for helping expand the use of Monolix and developing new functionality there. She'll talk about using a population PK/PD model developed in phase 1 to select a dose for phase 2, and with a specific example, which is increasingly common, of monoclonal antibody. She'll be followed by Cognigen's Yogesh Patel, Associate Director of Quantitative Clinical Pharmacology, and he'll be giving an overview of pharmacometrics in phase 1. So first in human and dose selection for phase 2. So Monika will be talking about a specific monoclonal antibody example, and Yogesh will be giving a bit of an overview. And then from our Lancaster division, we'll be having Mike Bolzer, who's one of the founders of Simulations Plus, and he'll be talking about GastroPlus and its DDI standards updates. So these are the 3 speakers that we'll be having, and then those will be followed by a series of posters, which are shorter presentations. Kyung-hee Yang, who is in our DILIsym division, headquartered in Raleigh, North Carolina, although I believe she's not there, perhaps in Texas. She'll be talking about assessment of the mechanism for remdesivir associated clinical ALT elevations using DILIsym, their DILIsym platform. And of course, as you all know, this is especially significant given the importance of remdesivir in treatment of COVID-19. She'll be followed by Vinal Lakhani to talk about quantitative systems toxicology and the mechanism and data gaps for evaluating hepatotoxicity of a new compound. And then finally, Lancaster's Bill Van Osdal will be talking about computational simulation of dermal absorption from complex topical formulations. So with this, I will turn things over to Monika, and I need to do a couple of IT things here first, and then we'll go from there. So